as you heard, this summer I had the opportunity to work at the Summer Research Institute here, St. Thomas University. My mentors are Dr. Jeffrey Plunkett and Dr. Tapanes Castillo. And I had the opportunity to work on the effects of neuronal adhesion molecule L1.1 over a growth-inhibitory chondroitin sulfate proteoglycans in a primary adult zebrafish brainstem cultures. So to give you a little background so you have a better understanding of the L1.1, which from now on I will refer to as the L1. The L1 is a transmembrane protein. So as you know, all eukaryote cells have a plasma membrane. And what the plasma membrane, it allows to separate the extracellular fluid as well as the machinery of the cell. So this transmembrane protein, which goes transversely the plasma membrane, allowed cell adhesion. What do I mean by cell adhesion? It will be kind of like if you guys come and stick together. So that's what cell adhesion means. What is its influence? It has a positive influence on neurite outgrowth. So that means it's involved in spinal cord regeneration in the zebrafish brainstem. And it was shown by scientist Thomas Becker that the influence of the regeneration. So as you can see here, this is the extracellular domain. These are fibronectin proteins, and these are immunoglobulin-like superfamily repeats. This is the intracellular domain of the protein. So what kind of interaction would I expect? I would expect a homophilic interaction of L1 protein, L1 and L1, and this interaction will allow outgrowth and regeneration as well. I would also expect other heterophilic interaction with the extracellular matrix, which can be the neurocam, that you will hear later on in this talks, and also with cell adhesion molecule known as CAM. So, my purpose of my project was to reduce the L1 protein level and analyze its functional relevance in CSPGs in vitro. I hypothesized that the L1 protein expression is essential for outgrowth in vitro over an inhibitory CSPGs around it. The question stands now, how do I go about reducing the protein level? What I used was the Morpholino. And the Morpholino is an antisense sequence that binds to the mRNA. As you all know, the central dogma of biology is that proteins, or the DNA, is transcribed into RNA and then translated into proteins. But since I don't want those proteins, I'm going to come over here with this antisense sequence and block the protein, block the mRNA translation, inhibiting the protein translation. But how can I get to get that antisense sequence into the cell? The way I administer in vitro is by an endopoder. So what the endopoder does is that it binds to the exoplasma of the cell. Once binding in the exoplasma of the cells engulf it, and it takes it down. So then, once inside, the cell permeabilizes and is released. So once the antisense sequence is inside the cell, that means, as I said before, that this sequence will recognize this mRNA and it will block protein translation. So the way I did my project was that I dissected brains and I processed this, as you have heard before. I incubated, and then after 2 days, I add the Morpholino and the endopoder. Then I did immunocytochemistry, as you heard before from Isaac. And then I saw it in the fluorescent microscope. So, the phenotypic response that I observed in the Morpholino-treated neurons were cells that were growing in that, I don't know if it's kind of difficult, this is a brain nucleide over here. I tried to do the best I could to show it. This is another cell here showing going into a CSPG's environment. Also crossing in the CSPG's environment, that it comes from here, the neurite, you can follow it. This is the CSPG's environment. Other cells being repelled. You cannot see well, but right here, the cell comes from here, the brainstem nuclei, and then once it encounters the CSPGs, the cell kind of deviated. It goes, okay, I don't like that. So cells that are growing inside the CSPGs and extending its axon outside the CSPGs. The CSPGs is in red, as you can see. So to have a better understanding of this, I asked myself, does the response to CSPGs vary between the control and the L1 morpholino? So a control morpholino is a sequence that will not recognize the mRNA. So if it does not recognize the mRNA, that means that the protein will be translated. And the L1 morpholino, it will recognize the mRNA, and I should expect that those the protein level should go down. So to have a better understanding, I analyzed this. I did statistical data of the phenotypic response of neurons, and this data was previously taken from the lab. So what I observed is that the morpholino downregulation of the L1 increases the percentage of cells inhibited by CSPGs. So here, in green, you can observe the morpholino, and these are cells being repelled, as well as cells with the control morpholino. The difference, we can see about a 16% difference if we look at this at 47% of the L1 morpholino, as well as the controlled or the non-treatment. And other cells that I saw that should be crossing in, that has the morpholino, are not, the downregulation it's significant from the non-treatment. And we find this a p-value of 0.1% chi-square in the non-parametric test out of 245 cells, meaning that this is statistically significant. Running more experiments this summer, I was able to observe that now, as I did more experiments, I was able to observe that there is no statistical difference here of cells being repelled of only 5%, and also cells of crossing in with the control morpholino. So I didn't observe no statistical difference, and its p-value was greater than 0.5% in a non-parametric test. So what I did to have a better idea, since these were raw numbers of cells that I saw, as you heard in Rachel from the cell study, I combined my data, and I observed to see if the cells repel, we're going to have a chance to show a difference or a significant difference, or cells that are crossing in to show another significant difference. I did not observe a significant difference on my statistical data. So I saw a p-value greater than 5% in a non-parametric test. So my conclusion is that cells with control morpholino and L1 morpholino responded the same to CSPGs. The caveat to this is that I still have to verify the knockdown of the morpholino inside the cells to see how it responded. However, Thomas Becker was able to show that using in vivo, he induced the L1 morpholino, and the fish took its axonal growth was not as other fish. It took a longer time for its axonal growth. So I plan to verify that knockdown. So my future directions is to repeat this experiment and also use the L1 protein as a substrate in primary neuronal cultures. Right now we're using laminin and cheat CSPGs with laminin. So this is kind of like an eagle view of the slide that we're using of the wells. And our future directions is to use the L1 protein and the NCAN. These NCAN will be taken from the fish, from the zebrafish, and L1 molecule. So as I said before in the beginning of my slides, there will be more homophilic interactions between L1 and L1, and we'll be able to see heterophilic interactions with the zebrafish NCAN. My acknowledgement, I would like to thank Dr. Plunkett, Dr. Alexis Tapani-Castillo for been working tirelessly, and Ma'am and Francis Alethea, they also helped us to work to get into this project, as well as my lab members, the U.S. Department of Defense, and the MS-EIP grant as well for paying us this summer. Any questions? Yes, sir.
Okay, you're trying to use hypothesis testing and you're using the non-parametric statistics. Could you tell me what you consider your null hypothesis and what you consider your alternative hypothesis in your study?
What I did is that since I don't have a way that I can say that these are the values that I would expect on the silver on the way of on the L1. I don't have a way to say, okay, these are the expected values. So what I do is that I make a column, and I see the numbers of cells, as you saw it in ratio. I see the numbers of cells that are being repelled, that are crossing in, that are crossing out. I use it, and then I run it through a test, through a computer test. It's instant gene, and it's able to tell me the significant difference between them of the cells that are crossing in or being repelled.
Okay, you might have a difference that could be significant depending on the size of the sample that you have. Mm-hmm. But if you don't consider properly the setup of the hypothesis, what might happen is that in average, you can see no effect at all. But you can see for a small sample just a very high dispersion of what you're getting, which sometimes it might work, sometimes it might not.
Yes.
And probably you're lucky to see the time when it works. So how you might discriminate between
well,
the two?
right here, as you can see this previous data, we found a p-value of less than 0.1%. So that p-value tells me that the chances of that the effect of this will be caused by the L1 Morpholino. In this one, it has a 0.5%. It could be by random. So that's why I said at the end that I still have to verify the knockdown of the gene, which we're going to do it, and then we'll have it. But it has been proven by Becker that the inhibition by the L1 is reduced. So we're doing it with the future directions and the L1 interactions with L1. Does it answer your question?
At some point you mentioned that you were going with antisense, right? So it means you knock something down. What guarantee can you give me that you did not change anything else in those cells with anything? I mean, if you do an antisense, can you promise me they didn't activate something else or you knocked something additionally down?
First of all, I cannot promise that.
Exactly my point.
You have been trained well.
Second of all, it was interesting because I was looking last night at a video, a gene tool video, and it shows the specificity of the morpholino and the way that it administered into the cell.
Right, but your first answer was fantastic, because what does that tell you about anti-sensing?
That of the recognition?
The anti-sense, yeah, what does it tell you? Because you said I cannot guarantee it. So what does it tell you, the technique?
The technique, well, the technique that I will use to verify now will be by Western blot, and to see how if the proteins are made.
That's not what I meant. I meant, what does it tell you of the technique of anti-sense? If you say I can promise it to you.
I'm sorry.
Because I said, you know, because my question was, use anti-sense, right? How can you guarantee me that you did not activate something else in the cell or knock something else down? If you start missing, for example, you have two hands, right? I got one hand off, and that's the hand you're writing with. You're going to be whining for a week, but pretty soon you're going to write with your other hand. So your ultimate pathway was activated. If I cut off both hands, I swear you're going to start writing with your feet sometime. It's going to take a while, but what I mean is, you knock something down. What's the danger is that was my question. How do you know, or how did you check that something else was activated or deactivated? That happens a lot with these antisense things.
Honestly, I'm going to have to revise more of that question. I don't know exactly how to answer.
In that case, Dr. Steelen, if you get the cellular response, then you know you can't say definitively that nothing else has been activated, but you know it's likely. I mean, you know, you're knocking down, and like he said with the Western blot, if you see that protein's knocked down, I agree with you. I mean, but the time frames of delivery and harvest are so close. I mean, you could go down a slippery road here. You could say, okay, well, you know, maybe it's minutes that it's activating something else. We don't know. We just don't know the answer to that question. But what we're looking for is a phenotypic result in the experiment, and that's the outcome.
Okay. Any other questions?
Any other questions?
I have a question. You said, well, we were all like studying if I understood correctly, you're all studying the like regeneration of cells in the zebrafish, right?
Yes.
So why is it that you were studying different proteins, or is it that you were studying the different proteins in the cells to see which
I'm
is
studying a
responsible?
gene, the L1 gene, to see the expression. If this gene it's upregulated once we do once there is once there is an injury in the site, this gene is upregulated, so could be possibly causing axonal growth regeneration.
And the proteins that you use are different from the proteins used in the other research.
What do you mean by that?
Yeah, the L1, and she said she used the Nepsin, right?
Nestin?
Yeah.
Nestin is more it's used in immunocytochemistry. It's more for recognition of proteins, yes, extracellular proteins. But what I'm doing directly is just knocking down a protein to see its expression, to see how the zebrafish in vitro would actually react, how the difference of repel and crossing in and crossing out. Yes, Professor.
Do you know where the name L1.1 comes from for your protein or your gene?
For the gene? Honestly, not remember.
Probably somebody found it along the line, along their research, and they just gave them names, and that was the one that they stuck with because that was working for them. So these names are usually don't mean anything except that somebody named them almost randomly,
Right.
or in a certain order. And then we keep the names, but then eventually, I'm sure if you find that it's really important for the system, you will receive a more sophisticated and honorable name.